Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 2.6, Facilitated Diffusion. We've talked about membrane transport as a whole, but now we're going to be talking specifically about facilitated diffusion and how the structure of a molecule affects its ability to pass through the plasma membrane. And so we'll be talking about membranes becoming polarized, about charged ions, different kinds of membrane proteins that allow for facilitated diffusion, and also aquaporins. And our suggested skill will be argumentation 6EB, predict the causes or effects of a change in or disruption to one or more components in the biological system based on a visual representation of a biological concept, process, or model. So if our membrane protein is removed, then what is going to happen to transport in and out of the cell? So as we've established in previous lessons, to transport large polar molecules or charged ions across the plasma membrane, you need integral or transmembrane membrane proteins, basically those that go through the entire membrane. And this type of transport is known as facilitated diffusion. It's also a form of passive transport. So basically, we're using membrane proteins to be able to transport polar molecules and large molecules across the membrane because, of course, large molecules can't fit through the gaps in the phospholipids and charged molecules are repelled by the phospholipid heads, the hydrophilic heads. So they require these proteins. Now there are a couple of kinds. First of all, we've got channel proteins, and this is just like it sounds. It's a channel, and it allows the passage of any substances that are the right size or the charge to go through. And this is primarily used for ions. So if we take a look at this picture on the right, this is the channel protein. It's literally just a tunnel which these substances can travel to from the high concentration to low concentration, so we're going with the concentration gradient. This is still passive transport. And one kind of these are known as aquaporins, and these are specifically for the transport of water in large amounts, because remember, in small amounts, water can make it through the membrane by itself. Next up, we've got carrier proteins, which bind to specific substances and move them across the membrane. These are primarily for molecules, so large polar molecules, or such as sucrose, or glucose, or fructose. These are going to be going through carrier proteins. Again, we're still going from a high concentration to a low concentration. It's still passive transport, but these molecules are being grabbed by the carrier protein, and they're being forcibly moved through across the membrane, and then the protein opens back up, and we get the next molecule to go through the protein. So, difference between channel and carrier. Now let's take a look at the role of membrane proteins in active transport. Membrane proteins are also used for active transport. When transporting things across against the concentration gradient, using, of course, ATP, ions and molecules to be transferred across the cell membrane still need membrane proteins, whether we're going with or against the concentration gradient. Now, here, there's also a new term we need to introduce, which is the membrane potential. This is the electric potential caused by the ionic concentration gradient across a membrane. So basically, the electrical charge on either side of the membrane due to ions. And we have this in addition to the chemical gradient, which is just the concentration gradient, which side of the membrane has more of the kind of ion. And together with this electrical charge, this membrane potential, along with this chemical gradient, we get what's known as the electrochemical gradient, which is going to affect the movement of ions both based on concentration, but also based on charge, how many negatively charged ions are going to be on one side of the membrane and how many are on the other. It's going to determine which side is going to have a positive charge or not. So over here in this diagram we can see extracellular, we've got more positive charge, while intracellular we have a more negative charge, but there are also these individual concentration gradients. For the sodium ion is going to be moving into the cell, the potassium ions are moving out of the cell, and the chloride ions are also moving into the cell. So that's just membrane potential. Now, probably the most famous and common type of active transport using a membrane protein is known as the sodium-potassium pump, and it might not be explicitly referenced on the AP exam or on your AP exam, but it will be a helpful diagram to know because there is a chance that you will get questions that do involve active transport and membrane proteins. So it is an active membrane protein transport using ATP, because this is active transport, to cause a change in the shape of the carrier protein and transport sodium and potassium across the cell membrane. So how it works is like this. First of all, the sodium ions are going to enter the carrier and we're going to have a phosphate group attach. So we split ATP. And once they've entered here, they're in place, this phosphate group attaching is going to cause a change in the shape. So all of a sudden they're going to open up out from the cell. So now the sodium ions can leave and the potassium ions are able to enter, which is what we see happening in picture 3. And then once the phosphate group leaves and goes back to join the ATP and detaches from the carrier protein, the protein goes back to its original form, and now the potassium ions can leave and enter the cell. So basically this is using energy to get one type of ion out of the cell and get another one in at the same exact time. This is the sodium-potassium pump, and it's used it's very important, used all throughout the body. And that's it for 2.7. Here's our facilitated diffusion review. 1. You should know about membrane proteins, carrier proteins, channel proteins, aquaporins. You should know they're used in passive transport, facilitated diffusion, and also active transport, namely with the sodium-potassium pump. So hopefully this was helpful. Next up we're going to 2.8 and learning all about osmosis.